Welcome, everyone. Thanks for attending today. So for anyone that's attending this call for the first time, we've been trying to hold this industry-wide seminar every month with basically any company that wants to contribute to the cultivated meat value chain. And so the purpose of these calls is to offer new and existing cultivated meat stakeholders an opportunity to learn from each other, especially with respect to some of the obstacles and potential solutions that exist along different points in the value chain. Ultimately, having an end goal that will accelerate R&D and commercialization in the sector. So if your group is interested in presenting on a future call, or perhaps you're working with a company that could benefit from being on these calls, do feel free to put them in touch with me directly and we can get them added. Also a reminder that you can extend the invitation to these calls so you're on our mailing list and you have the call details every week or every month by extending the invitation to your colleagues at your company as well. Before we begin, just a few housekeeping items from our side. So if you are a researcher that has worked on cultivated seafood, whether you're developing cell lines or long-term primary cell cultures from aquatic animals, we are interested in learning from you. And so we've assembled a survey that captures a lot of the questions that we're interested for a report that we'll put together that sort of summarizes the learnings and trial challenges to seafood cell line establishment. And so we have a survey, and if you can fill that out, if you do work on seafood cells, that would be really helpful for this project. Thanks, Renee, for dropping that in the chat. Sticking on the topic of seafood, so we also are holding our next Fish and Chips event on March 25th. This is just a virtual collaborative huddle that's hosted by GFI, and that aims to bring together different researchers from academia, industry, other interested parties around the world that can sort of share learnings on how to tackle some of the challenges with cultivated seafood, ranging from cell line development or media optimization, etc. And so we're having that event on March 25th, and the registration you can find in the chat. My cat is walking across my keyboard. And then lastly, I wanted to share the Advanced Regenerative Manufacturing Institute, or ARMI, has launched a joint call for commercialization resource proposals for biomanufacturing and health technology companies. GFI is a member of ARMI, and they're interested in cultivated meat and sort of incorporating a lot of the learnings that's happening in this field to other areas in cell therapeutics and regenerative medicine. And so essentially this call for proposals is to gain access to some of the services that ARMI offers. So you can view the full proposal details in the ARMI member portal, which I'll drop in the chat, and noting that both members and non-members are allowed to log into this portal to view the full proposal call, and you are welcome to submit proposals. I think the due date is in March, and so we're trying to get this word out to folks who might be interested in that. Alrighty, so that is all from my side. Today, we're pleased to be welcoming Adam Richardson from Panome Bio, who's going to be sharing more about how metabolomics can help inform and guide numerous aspects of your research. So Adam, I will turn it over to you.
Well, thank you so much, Elliot. Thanks everybody for joining and for having us here today. Yeah, we're happy to talk about our platform and continuing on the seafood theme, talk about some real world data and results out of a cultivated seafood partnership we did with Balloonaloo. So. All right, so I'll start with a little quick intro to Panome Bio, and then I'm going to touch just real briefly on our approach. I know a lot of these type of talks tend to get into the technology and lay the foundation, but I want to jump right into the results and talk about the work we did in this bioreactor with Balloonaloo, and then I'll circle back around and tell you about some of the technical details and kind of how we collected the data at the end. So my name's Adam Richardson. I am the study director at Panome Bio. We're a startup company, a couple years old, out of St. Louis. We were started as a collaboration between a number of people in St. Louis. One of them is Gary Patty, our academic founder here. He's at Washington University, departments of chemistry and medicine, and has developed some really nice technology over the last 20 years that serves as the foundation of what we do here at Panome. But he partnered a few years ago with a group of people who had launched some CROs before and done this type of work in the space. And working out of this BioSTL, which is an incubator biosystem or incubator system here in St. Louis that partners with Washington University. We've got access to some really nice shared lab space as well as our own lab space and instruments, and it's been a really great way to translate some really deep foundational science and technology and make it available to people. So Panome Bio we started really with that focus in metabolomics, but we are a multi-omics company at the end of the day. We work across the metabolomics, proteomics, and transcriptomics levels of the omic layers. We're really getting closer and closer to the phenotype as we move through these layers, going from genes to proteins to metabolites. Genes, of course, are the roadmap of what could and might happen in a particular cell or a particular system, but the proteins ultimately are what are doing the work, and the metabolites are the output of that work. So we believe that by measuring and then integrating these various layers of molecular signaling and function, we get a much broader, deeper picture of what's going on within your system, which gives you a lot more information about the points of control and where you can alter that system to optimize it for your needs. We do work a lot in the life science area as well, but really our tools and our technology apply across biology. They're very species agnostic, and we have done quite a bit of work in the cultured meat and the bioreactor space. It's been a really interesting application of what we do. So fundamentally, we are a mass spectrometry focused laboratory. This is our primary tool. It really enables untargeted analysis, and this is a combination of the power of mass spectrometers in 2025, along with the computational tools that are now available. When you try to do a truly untargeted molecular experiment and profile all of the metabolites or all of the proteins, your first round of measurements produces tens of thousands, if not over 100,000 individual signals within that mass spectrometry run. A lot of that is noise. A lot of that is redundant. There's a lot of signals to dig through to make sure you have correct, confident IDs and observations of individual small molecules, proteins, or transcripts. And that's what this platform and this technology, first developed in Gary Patty's lab and now extended at Panome, this is our core enabling workflow. But before we get into the real details, and I'll tell you a little bit more about how that works so you can understand what the options are with this sort of approach, I want to jump right into our real world data set. We have a long-running collaboration with Blue Nalu Seafood. They're working on optimizing their culture system, optimizing their production of seafood as a product. And we did some work with them to help them characterize their bioreactor system, both in terms of what's going on within the cells, what type of chemical and protein profiles they're generating, and what's occurring in the media, what's occurring in that culture feed. Are nutrients being depleted? Are they being overproduced? And we integrated those interpretations to provide a complete picture of what was going on and make some recommendations about what could be adjusted. As I'm sure many of you know, the basic experimental setup is Blue Nalu is operating bioreactors, feeding in media, and growing these seafood cultures, ultimately to serve as a food product. And we are running that through the mass spectrometer platform we just told you about, with no prior bias or targeted panel of the sorts of compounds we're looking for. Targeted panels are a great approach. They're very robust. They're just very limited in scope. If you already understand what's going on in your system pretty well, that can be some really valuable data. But when you're really looking to discover and understand that system from the ground up, an untargeted approach is going to make all of the possible measurements within this system and then put those back into one complete picture. So we worked with this system across 7 time points. I'll say it was a multiple day system, and identified metabolites that were significantly changed with high confidence in both the cells and the media within that system. And you can see here we detected thousands of metabolites in each system. We analyzed those for statistical significance based on fold change and p-value. And the spots in red are indicating individual metabolites that were altered with both a two-fold change and a p-value of less than 0.05 across this experimental system. So in the supernatant media here on the right, the red dots on the left side of this plot will be nutrients that were depleted from the media over the course of this growth, and the red dots will be things that were excreted from the cell. It's not surprising you see a lot more upregulated metabolites than downregulated metabolites because your starting media is relatively well defined. There's a certain number of metabolites in the media which will be depleted and consumed, but the cells can produce a little bit of almost anything. So you'll get a much wider variety of metabolites being produced and excreted by the cells. So we narrowed down these observations of a few thousand metabolites down to about 150 metabolites that were significantly changed across the system. But what we want to do, of course, is tie the cellular metabolic activity back to the media, because the media is what is most readily optimized or altered in order to adjust your system. And here's where we start to pair individual metabolites with their changes in both the cells and their media, and consider the magnitude of the fold changes across each system. We have these are just being reported here as metabolites 1 through 14. Within the confidential Blue Nulu report, of course, these are all fully identified metabolites and placed within their pathway context. But you can see that within the metabolites highlighted here, changing across these 7 time points, there are metabolites such as metabolite 2 or metabolite 4, which seem to be very directly linked between their cell and their media concentrations. The metabolite levels go up in the cells and they go up in the media. A good candidate for something being produced by the cell and excreted into that culture media. On the other hand, there's things like metabolite 9, which seem to be actively excreted from the cell. It's a waste product, perhaps, and the cell is trying to get rid of that into the media. The same thing with 10 and 11. Maybe it's something that, because of whatever is being fed into this culture system, is producing an excess of certain metabolites within the cell, and those metabolites are then being disposed of into the media environment. We then break out these changes into a number of heat maps and statistical analyses. You can see here there's about 60 metabolites that were significantly upregulated across this time course, going from left to right, with your latest time point on the right. And similarly, metabolites that are consumed. There's about 6 metabolites here which were significantly depleted from the media by the end of this time course. And then we move to the overlap of these systems to try to pin down which metabolites have the most functional support for their role within the cells. And you see some very distinct patterning here. The set of compounds, these 3 compounds here, they increase in both the cells and the media to a very similar extent at very similar time points. And once we start to add these layers of information, whether it's the media plus the cells, whether it's metabolites plus proteins, the more of these perspectives we have on the system, the more confident each of these individual conclusions are. And then one of our core tenets here at Panome Bio is that we don't want to just deliver you a list of 1,000 compounds that changed in your system and say, okay, here you go, good luck with the interpretation. We do want to put these in a biological context and use our experience and expertise to help you translate all of these molecules into some type of functional output. And one of our big tools is pathway enrichment. So this will start to ask now, of all of these individual compounds that we see altered across your system, do they cluster within specific pathways? And on average, are individual biochemical modules up or down regulated as a whole? And that's what we get into here. So these will be either individual biochemical pathways such as glycolysis, fatty acid oxidation, amino acid biosynthesis, or they may be classes of lipids. We may have the observation that say, hey, your phosphatidylcholines are universally upregulated at these later time points. So there's something interesting happening around fatty acid biosynthesis and maybe membrane fluidity. And that's what we try to do, is put all of these individual molecules into a broader context. And again, layer on the observations. So we're really not pinning too much weight on any single molecule. We want to understand what's happening in the system as a whole. And ultimately, we are reporting both the up and down regulation of individual pathways, as well as how many metabolites in that pathway were individually significant. And here are some of the conclusions and the interpretations that we provided to Blue Nalu at the end of the study. This is based upon both the cells and the media, and there was the observation that the media seemed to contain an overabundance of a specific nutrient. It was overwhelming some of the cellular biochemical pathways. Separately, there was amino acid that contributed to the same pathway, and the combination of these two inputs really drove a lot of downstream biochemical activity, which shifted some of those molecular pathways, right? You can see things like a shift away from respiration towards glycolysis. And then our interpretation around these biochemical modules led us to conclude that the bioreactor appears to be oxygen deficient at the later time points. And based upon these conclusions and these molecular profiles, we did make some specific recommendations to Blue Nalu around modifying the levels of those media components to bring them more in balance with the other inputs of that pathway, as well as increase either the oxygen levels or the mixing in order to keep the core biochemical activity in those pathways turned on or off at the optimal state across that time range. I'm going to circle back around and talk a little bit about metabolomics here. So metabolomics has meant a number of things over the last couple decades. When we talk about metabolomics, we really imply polar molecule, lipid molecules, all of the small molecules within your system between 50 and 1,500 Daltons. We come at this from a truly unbiased, untargeted approach, from the sample preparation to the mass spectrometry measurements to the bioinformatic analysis. So we don't have any type of chemical class bias. We're not working with targeted panels. We're trying to capture the system as a whole. And obviously, cells and media are the most relevant samples, I think, for this group for a bioreactor system. But I will note that this is very species agnostic. You know, that's why we could work across a seafood culture, a beef culture, as well as things like feedstocks. So I like to say that if you can put your biology in a tube, we can measure the chemistry. And this is this a targeted approach that I've been talking about. 20 years ago, computational power was much more limited than it was now. Everyone was working on desktops. That's about it. And when you're trying to do an untargeted mass spectrometry experiment and get tens or hundreds of thousands of signals, it's like putting a puzzle together where you're not sure what the picture is supposed to look like at the end. So the easiest way to get that done is to simply try to match your puzzle piece against a previous known puzzle piece. And those would be all of the standards that you ran in your system. And that's a great start. You're limiting your search space to something you can accomplish. But now we have the computational power to make hundreds of thousands of those searches at a time rather than merely dozens. And that's one of the core capabilities of Panome Bio. And that is our ability to ask what has been reported by humanity in the public databases. Is there a match for your compound? Is there a function for your compound? So we bring all of that data in and compress it down and apply it to your particular experiment. This untargeted discovery approach is great. As I mentioned, by default for us, it covers polar metabolites as well as lipids. We can separate those out, absolutely, if that's a way to be a little bit more efficient with your experiments. We do have those quantitative targeted metabolomic panels, if that is what is needed for your particular project. We can do some really interesting work with metabolic flux and stable isotope labeling. Maybe you've done some work and you know a particular pathway is up or down regulated and you want to learn more about exactly how that pathway is controlled. Well, we can feed those cells a stable isotope labeled precursor to your compound. Stable isotopes, of course, are not radioactive. They're perfectly safe to add in any environment. But then when we do the metabolomics, we'll be able to tell you not only was your compound of interest changed up or down, but how much of that compound was biosynthesized over those last 24 hours since you added the feedstock. And you really start to get some deep understanding of the biochemical activity within your cells beyond just the metabolite levels. All of this applies perfectly well to our proteomic approaches in addition to the metabolome. Certainly the proteins are of relevance due to their enzymatic capabilities and the fact that they're often driving a lot of the cellular activity. And depending on your context, expressing certain types of proteins may also be critical for the value of that final product. So we do a mass spectrometry-based proteomics approach as well. We have a really nice sample prep method for spent media using the sear proteograph system. It uses nanoparticles to enrich your low abundance proteins, and you can start to pick up growth factors that your cells are secreting and signaling to each other and possibly controlling your final output as much as the metabolite input. And because this is just a true unbiased observation, we pick up things like PTMs and proteoforms as well, phosphorylation, glycosylation. These are all things that affect not only protein function, but the experience of consuming it as well. Here's a little picture of that sear proteograph for our culture medias. Also works very well on serum. If anyone does further downstream work about nutritional inputs and how that impacts a human or an animal. And we tend to report 5 to 10,000 proteins per sample. It's really top of the line, deep proteome coverage with this combination of the proteograph and the new Astral mass spec from Thermo Fisher. One of the big challenges with mass spectrometry is doing longitudinal experiments. We have a very well-developed reference approach where we take all of the samples in your particular project, pool them together, and use that as a QC to do some technical corrections around signal alignment. But then if you have a follow-up project down the road, we'll rerun that QC sample, and that lets us integrate all of your data and results across these experiments across time. And then ultimately, as I mentioned up front, we are a multi-omic company. Metabolites are great. They tell us a lot about the function. But if you start to build up multiple layers of omic data here, metabolomics, proteomics, transcriptomics, and this doesn't necessarily have to be generated by Panome. You may already have some of this data in-house. We can most likely integrate your existing data sets as well. And this everything that we find altered or operating at multiple levels really just increases the confidence that that is truly a functional module within your system that might be a point of control. I've already shown you many of the data outputs. We do put a lot of work into our interpretation, our statistical analysis, and our contextualization of the results. We don't want you to have to sort through hundreds or thousands of molecular IDs on your own. So we always do a follow-up meeting with clients. If they wish, we'll walk them through all the data, explain it to them, make any adjustments, answer any questions. So we really try to be fully engaged and make sure that we execute the experiment in the way that delivers what you're looking for. And as I mentioned, this is about a 4 to 6 week turnaround time. We receive frozen samples shipped to us on dry ice. We'll do the sample prep, the measurement, and the reportings, and you'll get back all the data layers, a nice method section that explains what we did, how we did it, and really tries to put all of the value back in your hands. So I'm happy to take any questions. You can find us at panomebio.com. We have a lot of resources here. This Blue Nalu example report, as well as a large number of others, are at panomebio.com/resources. So if you have interest in anything else, feel free to reach out by email or look at our website and see if you can find something of interest. I'm happy to take any other questions.
All right. Well, thank you again so much, Adam, for the presentation. And yeah, feel free for folks to get in touch offline. And yeah, we'll see you again here next month. Thanks, everyone.